This is part 2 of chapter 4. We're talking about virus replication cycles, and we've gone through some of the early steps: attachment, penetration, uncoding. Step 4 in the replication cycle is replication. It's kind of confusing, but this is referring specifically to replication of the genetic material, DNA or RNA. Um, and so, yeah, this is just copying the viral genome, right? And the way in which this happens is going to depend on what the genetic material is, right? Is it double-stranded, single-stranded, DNA, RNA, etc.? We'll go through some of the different types here in a minute. The location in which replication happens inside the cell may vary. A lot of times it may have to occur in the nucleus because that's where a lot of the enzymes for nucleic acid replication in the cell may be, but some viruses will encode their own enzymes to replicate their own genome. So that may vary quite a bit. Viral genomes can be what are called segmented or non-segmented. Non-segmented would mean the genome is just a single molecule, whereas segmented means you have multiple DNA or RNA fragments that make up the genome. Genome could be linear or circular, so that could affect what replication looks like as well. And you may remember in chapter 2, I mentioned the Baltimore classification system, which primarily, but not exclusively, categorizes viruses based on their genome type. We have these 7 classes: double-stranded DNA, single-stranded DNA viruses, double-stranded RNA viruses, and then we have plus or minus single-stranded RNA viruses. What this distinction means: a plus-stranded RNA is basically equivalent to a messenger RNA in a cell, and that could be immediately translated into proteins, whereas a negative sense, you would have to make a positive sense complementary copy first before you could carry out translation. And then we have either RNA or DNA viruses that go through a reverse transcription step. So this chapter actually spends a lot of time going through some of the details of how these 7 different groups of viruses carry out their nucleic acid replication. And so right now I am just going to focus on group 1 here, the double-stranded DNA, but I will include an additional lecture where I explain and go through what the book has to say about replication for some of these other groups as well. So I encourage you to take a look at that, have a listen to that. All right, so just a note on RNA viruses, right? We had quite a few categories here that were viruses with RNA genomes. You may be aware of this, but RNA viruses are much more prone to mutation, which means they will evolve typically much more rapidly than DNA viruses. And why is that the case? Well, DNA is going to be copied by DNA polymerase, which you may remember has proofreading capabilities, right? If a DNA polymerase makes a mistake while copying the DNA molecule, it can usually recognize that mistake and correct it. Whereas RNA polymerases, which would be used to copy RNA viral genomes, typically do not have any proofreading capability whatsoever. So there's going to be a much higher error/mutation rate, which is going to cause these viruses to change very rapidly. And this is one of the reasons we see, like with the influenza virus, right? It mutates very rapidly, so you have to get a flu shot every year because there's going to be new strains constantly arising that previous vaccines may not be effective against. So we're kind of in a situation right now with COVID-19, because it is an RNA virus, trying to understand whether we're going to be in the same boat with COVID or not. If we can develop an effective vaccine, how long is that going to be effective? Might be a situation where you have to get a new one every year, but hopefully not. So we get bottom line is we get a lot of diversity in these RNA viruses due to that higher mutation rate, due to the mistakes made by the RNA polymerases in copying the genome, but there are a couple of other methods I wanted to point out that can also increase diversity in RNA viruses. So one is recombination, and this could occur if you have two different strains, basically two different genetic variants of one virus that have infected the same cell. And when the RNA polymerase, called RDRP, RNA-dependent RNA polymerase is what that stands for, when the RNA polymerase is copying the genome, it may sometimes jump from one strain to the next, basically back and forth. And in doing so, it will then create a new hybrid genome, right, that has portions of either parent strain combined into one. So that could significantly increase genetic diversity. And then you also have this phenomenon called reassortment. This would only apply if you have a segmented virus, meaning the genome consists of multiple molecules. So if you have two strains of a segmented virus infecting the same cell, when that virus is assembling new virions at the end of the infection cycle, it has to make sure it gets the correct number of RNA molecules into that virion. And if you have two different strains in the same cell, it may be picking, molecule 1 of the genome, it may pick from one strain, molecule 2, it may pick from the other strain. So again, you end up with this mixed genome that may be something new. So this phenomenon in particular is a big concern with the influenza virus, and we'll talk about that in a later chapter. But bottom line, RNA viruses are going to mutate more quickly, they're going to evolve more quickly, going to tend to be more diverse. So as I mentioned, we have these 7 different Baltimore classifications, 7 different viral classes, each with unique strategy for replicating its genome. But as I mentioned, I'm just going to focus right now on class 1, just to try to keep the flow of the lecture going. But I will have a separate lecture where I go through the other 6 classes for you, at least in some detail. All right, so let's look at these double-stranded DNA viruses. This includes some well-known human viruses: herpes simplex virus, Epstein-Barr virus, HPV, pox viruses like smallpox would fall into this category as well. And, we're familiar with double-stranded DNA as genetic material, right? Because that's what most life on Earth has. And, of course, double-stranded DNA is copied by DNA polymerases. And if you've ever studied DNA replication, you've seen that process before. And then RNA polymerases would be used for transcription. So because all the machinery in most host cells for DNA replication, for copying DNA, is in the nucleus, the majority of double-stranded DNA viruses are going to have to get their DNA into the cell's nucleus to be able to utilize that machinery for the copying process. Really, the one big exception to this rule are the pox viruses, which interestingly encode in their own genome all the enzymes and proteins that they need for not only transcription but genome replication. So they're a bit more self-sufficient, don't need to enter the nucleus. If we look at the little flow chart here, so we've got our double-stranded viral genome that has to be transcribed into messenger RNA. The messenger RNA has to be translated into proteins. This would be production of viral proteins. And then we also have to make copies of the viral genome. Once we have those viral proteins and we have copies of the viral genome, we could begin to assemble new virions. Because remember, the basis of a virion is the genome, right? Double-stranded DNA in this case, surrounded by capsid protein and then likely some attachment proteins as well. So typically what you'll see is transcription and translation happening first, even though it looks like they're simultaneous here, happening before replication. And usually that's because some of the proteins required for replication may be encoded by the viral genome. So, some of these proteins are coming from the host cell, but we'll very likely need some of these proteins to come from the viral genome to complete this replication process effectively. So if we're going to start with transcription, we're going to need transcription factors that are going to bind to promoters of genes in the viral DNA genome, recruit RNA polymerase, and initiate that transcription process. And very commonly, you will actually see these like waves of transcription of viral genes. So you'll have what are called the immediate early genes that get transcribed first, then followed by transcription and translation of the early genes, followed by transcription and translation of the late genes. And so the way this would work is, so you would get the immediate early genes transcribed, right? Those mRNAs leave the nucleus, go get translated at ribosomes in the cytoplasm, and those would almost entirely all encode, those genes would almost all encode transcription factors. So those transcription factors that are made would then go back into the nucleus, bind to a second set of genes, the early genes, their promoters, and activate their transcription, and so on. So we get these waves. And the early genes are usually going to include transcription and translation of proteins that are important for replication of the viral genome, whereas the late genes would be maybe more structural proteins like capsid proteins or some of those membrane glycoproteins, attachment proteins, those kinds of things. And the reason for this these waves is to get some temporal separation, right? You could imagine there could be an issue if the virus immediately started making capsid proteins and membrane glycoproteins, because the virus is not particularly smart. If it has those proteins, it's going to try to start assembling new virus particles inside the host cell. And if it's trying to assemble virus particles, but it has not yet replicated its genome, it's going to make non-infectious particles that do not include copies of the viral genome, which would be a problem. So we want to make sure that we get replication of the genome before we start assembling viral particles is the bottom line. And so in terms of how the actual copying of the DNA is going to take place, it could be very similar to what you've already learned if you've studied cellular DNA replication, where you have primase making a primer, then that primer being extended by a cellular DNA polymerase. That would be pretty typical, right? That's why the viral DNA is going into the nucleus, is to take advantage of those enzymes. It would be like, for example, herpes virus does this. But you do have some exceptions, like adenoviruses actually encode their own primer, which DNA polymerase can use to extend from and copy the DNA. Sorry. All right, so again, taking this back to this idea of assembly, right? We get transcription and translation of some of these proteins that we need for replication. We kick on the replication process. Once we've got that going, then we can actually start making some of the structural proteins, capsid protein, and then once we've got everything we need, we can finally start assembling new virions. And so that takes us into the fifth step, which is assembly. And this could look pretty different depending on which virus you're talking about. Could occur at different places inside the cell, might happen in the nucleus, in the cytoplasm, at the plasma membrane. It really just depends. But we'll see some specific examples in later chapters as we're talking about individual viruses of what the assembly process looks like. But it is typically very efficient, right? So you can see here a cell, it's infected with rabies virus, and you can see inside the cell these new virions, rabies virions being assembled, and they're really packing the cell pretty full, right? So it's happening at high efficiency for sure. Then that takes us to maturation. Maturation is referring to actually creating an infectious virus particle. So a lot of times you may have the virion that's initially produced is quote unquote immature, would not be able to infect another cell. So there would have to be some changes before it became infectious. One example is influenza virus, right? You might remember when I was talking about fusion, I was talking about the HA proteins on influenza virus and how they interact with the endosomal membrane. Well, it turns out the initial influenza virion has this immature HA protein that has to be cleaved by a protease into the HA1 and HA2 proteins. And until that cleavage happens, the influenza virion has not matured. So in this case, this is happening by a protease inside the cell. Another example would be HIV, famously. So HIV assembles new virions at the plasma membrane. It buds out of the plasma membrane. We'll talk about that process in a second. And even though it has left the cell, it is still immature. So you have what's called this GAG polyprotein that has to be cleaved by a viral protease. So HIV actually produces its own protease. It's one of the proteins that it packages up in the virion, in the virus particle here. So that HIV protease will cleave the GAG polyprotein into capsid matrix, nucleocapsid proteins after the particle has already been released. And then we get a mature HIV virus. And so there are quite a few protease inhibitor drugs that are used to treat HIV and prevent HIV spread that are acting specifically by inhibiting that maturation step, by inhibiting that protease. So in terms of release, getting the virus out of the cell, it may happen by budding, this budding process that I was just talking about with HIV. And so in budding, you will have all the viral proteins, the nucleic acid that are being produced inside the infected cell accumulated at a specific region of the plasma membrane. And as things accumulate, right, the plasma membrane is budding out and will eventually completely bud off from the infected cell. Sometimes budding will actually happen inside the cell, so it could happen in the plasma membrane, but we'll see some examples where we get viruses that actually bud through membranes inside the cell, through the rough ER, through the Golgi, for example. And you can see a couple of images here. This is rubella virus budding off of an infected cell. On the right here, we've got Ebola virus, which has been colored blue, budding out of an infected cell. And you can see there's just an astounding amount of virus, right, budding from a single cell. So in addition to budding, you could also get released just by lysis of the cell. So sometimes virions will accumulate to such a high level inside an infected cell that the cell will actually burst open, killing that cell. Or it could happen by exocytosis. The main distinction between exocytosis and budding would be in exocytosis, usually the virion would be inside a vesicle inside the cell. Then that vesicle is going to fuse with the plasma membrane, releasing the virion. So to finish this out, I just wanted to talk really quickly about virus growth curves, because this really ties in with this, with the whole replication process, and there's some important terms here. So in this graph on the x-axis we've got time, on the y-axis we've got the number of virions that are present, and there's some terms here like latent period, eclipse period. So the eclipse period would be the time it takes to form infectious virions internally. So we have two lines on this graph. The blue line is what's happening inside the cell, and the orange line is what's happening outside the cell. So in the eclipse period, that's basically all the time it takes the cell to go through, attachment, penetration, uncoding, nucleic acid replication. At the end of the eclipse period, we have started to generate virions inside the infected cell. And we see those numbers then starting to increase in the cell. When we hit the end of the latent period, that is where release is going to start happening. So some of those virions are being released from the infected cell, and then we see the number of released virions starting to increase at the end of the latent period. And so this may vary, obviously, from virus to virus. Every virus would also have what's called a burst size, which is how many infectious virions are being released from an individual cell, and that number may be incredibly high. So, okay, thanks for hanging with me through Chapter 4. And as I said, do check out the additional lecture that I'm going to post up that talks about nucleic acid replication for those other 6 Baltimore classifications of virus.